Hello students. So today we are going to learn the third part in cell structure and organization. In this part we are going to learn about vacuoles, glyoxysomes, mitochondria, plastids, ribosomes, nucleus and cytoskeleton, and also some of the animation images related to these topics. And in the first page, as usual, you can see cell structure, where you can see revolving cell, in which you can see the nucleus very clearly, the endoplasmic reticulum, mitochondria, the lysosomes and few other structures which you can clearly see in this. Now coming to the next slide, cell structure. As we learn the previous part, or we can recap the previous ones, and in this chart we can again see the entire topic in this single slide, where in prokaryotic cell we already learned cell envelope, glycocalyx, cell wall, plasma membrane, cytoplasm, in that mesosomes, chromatophores, inclusion bodies, ribosomes, nucleus, plasmid, pili, flagella, right? Whereas in the second type, eukaryotic cell, the first six we learned in the second part of the video or cell structure organization, that is cell wall, which in case of plant cell, plasma membrane or cell membrane or biomembrane, cytoplasm, cell organelles, endoplasmic reticulum, Golgi apparatus and lysosomes. Whereas in today's part we learn the further components, that is vacuoles, microbodies, pyrosomes, peroxisomes, glyoxysomes, mitochondria, plastids, again three types: leucoplast, chromoplast and chloroplast, then ribosomes, nucleus, cytoskeleton, in which we learn about cilia and flagella, centrioles and centrosomes. Now we learn in detail about the individual organelles, that is starting with vacuoles. Vacuoles are membrane bound sacs prominently found in plant cells. Now as you have seen the image of plant cell and animal cell, very clearly you can see the nucleus which is present in the center, very prominent, whereas in case of animal cell it is smaller in comparison. Then in animals cell, whenever present, they are few in number and smaller in size. Generally there are two or three permanent vacuoles in plant cell. As we have seen the permanent vacuoles in plant cell, which is very prominent, present in the central part. In some larger plant cells, a single large vacuole occupies the central part of the cell. It is called as the central vacuole, which we very commonly see in an image of plant cell. In such cells, vacuole can occupy as much as 90% of the total volume of the cell. Again, as you seen the image of the plant cell, the vacuole, which is single and large, occupies a major part of the cell, and the remaining parts, whether it is nucleus, chloroplast, they occupy the side region or the peripheral part, right? Okay, further, the vacuoles are bound by semi-permeable membrane called tonoplast membrane. This membrane helps in maintaining the composition of vacuolar fluid, the cell sap, different from that of the cytosol. Now, as we learned cytosol in the previous video as well, the cytoplasmic movement, the different organelles or components present, how it may move in the cytoplasm or cytoplasmic pool. Further, composition of cell sap differs in different types of cells. What does that mean? So every cell may have a different composition of cell sap. The components may be same, but the composition of the cell sap may differ. The cell sap of central vacuole is a storehouse of various ions, and thus it is hypotonic to cytosol, means high in concentration. Small vacuoles in seeds or certain plants store organic materials like proteins. Vacuoles store excretory products or even compounds that are harmful or unpalatable to herbivores, thereby protecting the plant. So as we know, some plants may be poisonous or it will be not able to consume. Why? The reason is the vacuoles may have got excretory products or certain harmful compound which cannot be consumed. If consumed, may have a bad taste in the mouth, or even it may affect. Further, Attractive colors of petals are due to storage of such pigments in the vacuoles. Now we can come to know why the petals are attractive or where these pigments are present due to which the petals are colorful, correct, in the vacuoles. Intake of food or foreign particles by phagocytosis involves formation of food vacuole. In fresh water, unicellular forms like paramecium, excretion and osmolation takes place by the contractile vacuole. Now I put a slide of paramecium further, which you can see where the vacuoles they take place in the or they help in carrying out excretion or osmoregulation. Hence they are termed as contractile vacuoles. Vacuoles maintain turgidity of the cell. Now here we can see the images. Firstly, I put the image at the top, animal and plant cell, only pointing out the vacuole, so it will be easier for to understand rather than all the labelings. So here you can see right side plant cell, which is quite bigger in comparison, one single almost in the central part, whereas animal cell the vacuole is comparatively smaller. Now again below the image of the animal cell you can see the paramecium unicellular component. We can see the contractile vacuole very clearly. Even the as it is animation part, you can see how the movement takes place inside the cell, that is paramecium, whereas right side you can see the movement in case of the plant cell, or as we use the term cytosol or cytoplasm movement, which is again very clearly seen in case of the plant cell. Now moving further, little bit of additional information here. In addition to endomembrane system, this we have learned in the previous video very clearly, or part 2 of the cell organelles. There are several other cell organelles bound by single layer of plasma membrane in these cells. Now whatever we learned in the previous video, that is about endoplasmic reticulum, Golgi complex, lysosomes. Other than that, there are few more. Which are those? Those are microbodies. Microbodies are found in both plant and animal cells. These are minute membrane bound sacs. Microbodies contain various types of enzymes based on which they are classified into different types, few of which are explained here. So here in short we are learning about spirosomes and peroxisomes. So spirosomes, these are found mainly in cells involved in synthesis and storage of fat. So what does spirosomes do? Storage of fat and also synthesis, that is production we can say. Endosperm of oil seeds, membrane of spirosome is half unit membrane, that is this membrane has only one phospholipid. Peroxisomes contain enzymes that remove hydrogen atoms from substrate and produce toxic hydrogen peroxide by utilization of oxygen. At the same time, peroxisomes also contains enzymes that convert toxic H2O2 to water. Conversion of toxic substances like alcohol takes place in the liver cells by peroxisome. So even the toxic components are converted into water, or they are converted into simpler forms with the help of the peroxisomes, which takes place in the liver. Towards the right side you can see the image where you can see this crystalline core, the central part, the plasma membrane and inner lipid bilayer. And also towards the right, as we saw this animation part in the previous video, oleosomes from smooth endoplasmic reticulum, also known as spirosomes, or the vesicles, transport vesicles, which you already seen. And below we can see anatomy of peroxisomes. When you make this section, you can see the inner lipid bilayer, plasma membrane and the urate oxidase crystalline core, which is in the central part. Now, in out of the different microbodies, peroxisome and spirosomes, we learn little bit about the glyoxysomes in detail. These membranes bound organelles contain enzymes that convert fatty acids to sugar. Now we have seen in the spiroxisomes they synthesize and store the fats. Here they convert fatty acids to sugar, that is in case of glyoxysomes. They can be observed in the cells of germinating seeds, where the cells utilize stored fats as source of sugar till it starts photosynthesizing on its own. So you can see the image in the left and right. Towards the left, we can see mitochondria, the glyoxysome, ribosomes, etc. Glyoxysome hydrolyzes fatty acids in the plant cell, and towards the right, the image of the glyoxysome, where you can see outer membrane, the matrix substances or the cytoplasm in it, and the central core part. Then, now coming to the next part, that is mitochondria. Now mitochondria, as we all know, it is a powerhouse of the cell. So you can see two images. Onto the left, the first mitochondria moving animation, and towards the right part, the transverse section of mitochondria, where you can see the outer membrane or even the inner membrane which is highlighting, and the cristae, that is the folds. So we now take the points one by one. These are important cell organelles involved in aerobic respiration. What does aerobic respiration means? Respiration taking place in presence of oxygen. Mitochondria are absent in prokaryotic cells and RBCs. So, as we learned about prokaryotic cells in the first part of the cell organelles, where mitochondria is absent but is present in eukaryotic cells. Their shape may be oval or spherical or spiral strip-like, which you can see presently. It is double membrane bound organelle. Again, you can see the outer membrane and inner membrane. Outer membrane is permeable to various metabolites due to presence of a protein which is called porine or Parsons particles. Inner membrane is selectively permeable to few substances only. Both membranes are separated by space. Outer chamber. Inner membrane shows several finger-like or plate-like folds which is called cristae, which you can see in the right side of the image very clearly, the finger-like projection, that is cristae. Inner membrane bears numerous particles, that is oxysomes and cytochromes, which are the electron carriers, which we learn in detail in the topic respiration. Inner membrane encloses a cavity inner chamber containing a fluid matrix. Matrix contains few coils of circular DNA, RNA, 70 S types of ribosomes, lipids, and various enzymes of Krebs cycle and other pathway. Where in Krebs cycles and other pathway we learned in detail about how the functions are carried out. Now, you can see here mitochondria, the inner component, much more in detail. Okay, so first you see the oxysomes. So what are the oxysomes? Just now you saw the folds, cristae, right, in which these oxysomes are present. So oxysomes, inner membrane of mitochondria bears numerous particles called oxysomes, which are F1 particles, F1, F0, which is Fernandez and Moron elementary particles, or mitochondrial particles. Each particle consists of head and stalk, which you can see towards the right end, yellow in color. You can see the cristae folds on which the F1 particles are present, and F1 particle, again you can see head, the pedicel or the stalk part, and the basal part. Further, head, that is F1, it appears like a lollipop, like orifice and lollipop, rounded structure. So you can see it's something similar like that, correct? Head faces towards matrix, and foot F0 is embedded in the inner membrane. Head acts as an enzyme ATP synthetase and foot as a proton channel. Oxysomes are involved in proton pumping and ATP synthesis, or we can say production of ATP. Now you can see the image of mitochondria, again the different parts in detail, the inner membrane, the inner membrane space between the outer and inner membrane, the matrix substance, ribosomes, cristae, DNA. Cristae, again, which has got the F1 and F0 particles, helps in ATP synthetase, where again towards the right, as I showed, the higher magnifications of the oxysomes and the cristae, whereas again you can see structure of inner membrane of mitochondria, that is single fold of cristae, where you can see the outer chamber, protein layer, lipid layer, intercrystal space that is between the cristae, F1 particles, respiratory chain and enzymes, mitochondrial crest, F1 particles, inner membrane, and the outer membrane. Now, as just now we read, F1 particles are present or projecting outward, whereas inner membrane, that is F0, is embedded into the inner membrane. Okay. Now coming to the next one, that is plastid. Now plastid, all of you know, that is chloroplast, more familiar with, but other than this there are few more components present. Which are those? We'll take one by one. Like mitochondria, plastid two are double-walled organelle containing DNA, RNA and 70S. So we could see the same point, same component or organelle present in mitochondria also. Which one? DNA, RNA and 70S ribosome, and also these are double walled. Only thing is component like chlorophyll or chloroplast, they are present only in the plant cell. Okay, further, but they are larger in size and can be observed under light microscope. Plastids are classified according to the pigments present in it, means the coloration or the pigment which helps to give the coloration: leucoplast, chromoplast and chloroplast. In the next slide we can see the all the three different types also in detail about the chloroplast. Okay, first we take leucoplast. That's why I highlighted leucoplast with purple color. Do not contain any pigments. They are of various shapes and size. That means leucoplasts don't have any pigment, but they have different shapes. These are meant for storage of nutrients. What are the main functions? Storage. Amyloplasts store starch. Elaplasts store oils. Oleroplasts store proteins. So you can see in leucoplasts there are three types: amyloplasts, which store starch; elaplasts store oils; and aleuroplasts store proteins. The second type, chromoplast, contains pigments like keratin and xanthophyll. Now keratin, xanthophyll we come across more common, or if I give the fruits and vegetables which are orange color, yellow color, they are mainly because of the chromoplast, or you can say keratin and xanthophyll. So they impart red color along with yellow or orange color to flowers and fruits, just as I mentioned. Now you must have understood why potato looks white in color and shoe flower petals are red. So we can easily understand now the content. Potato is white, reason it may have leucoplast, whereas petals are colorful because it may have chloroplast, or we can say chromoplast. In fact, not chloroplast, chromoplast, that is keratin and xanthophyll, right? Okay, so both leucoplast and the amyloplast do not contain photosynthetic apparatus. That's why which we find in chloroplast. Plant cells contain algae and few protists like euglena which contain chloroplast. Now chloroplast we can easily identify by the green coloration, but even in those there are differentiation like blue green, olive green, etc. Even though it is green color, but even there are different shades of green. But in case of chromoplast, it is moreover red, yellow, orange. As I gave you the example, the fruits, flowers having different coloration. Correct? Okay. Further, you have observed ribbon-shaped chloroplasts in Spirogyra. That is the algae. That means there are different shape of chloroplast as well. It differs in size, number and shape in various cells in which it is found. In plants, it is found in green regions, mainly in the mesophyll of the leaf. The chloroplast is lens shape, that is almost ovals like, but it can also be oval, spherical, discoid or ribbon like. So you can see different shapes. As we just said, Spirogyra, they are ribbon like, but other than that it can be oval, spherical or discoid, means disc shape. A cell may contain single large chloroplast as in Chlamydomonas, or there are 20 to 40 chloroplasts per cell seen in mesophyll cells. Chloroplast contains green pigment, that is chlorophyll, along with other enzymes that help in production of sugar by photosynthesis. So all of you know chloroplast, which helps in carrying out photosynthesis, which further help in the production of sugar. Now this we again learn in the chapter photosynthesis. Like mitochondria, we learn in detail in the Krebs cycle or in the respiratory cycle, whereas photosynthesis we learn in detail in the chapter about different cycles in case of photosynthesis in detail, that is cyclic, non-cyclic, Kelvin cycle, etc. Okay. Further, inner membrane of double-walled chlorophyll is comparatively less permeable. Inside the cavity of inner membrane, there is another set of membranous sac which is called as thylakoids. Now, what are these? We'll see in the next slide. Now, here we can see the thylakoids. First, we'll cover up the remaining points. Thylakoids are arranged in the form of stacks called grana. You can see stacks, or you just imagine coins kept one above the other. So, how when you arrange coins one above the other, how they appear, right? The grana are collected or connected to each other by means of membranous tubule called stroma lamella. That is, even this thylakoid which has got stacks called grana, these grana are also interconnected with each other, which you can see it can be called as intergranal lamellae or stromal lamellae, etc. Space outside the thylakoids is filled with stroma, or we can see the cytoplasm present in the chloroplast. The stroma and the space inside thylakoids contains various enzymes which are required or essential for photosynthesis. Like other plastid, stroma of chloroplast contains DNA and ribosomes. That is why they can carry out the various activity on their own because of presence of DNA and ribosomes, which again present in mitochondria. So you can see towards the left, chloroplast structure, ultrastructure of chloroplast, where you can see the section of chloroplast. Inside we can see red colored granule, yellow colored granule, green colored granule. The red one are the sugar molecules, and you can see the inner membrane. The other yellow color, we can say the other components present other than the starch component present in it, or we can say the sugar molecule present in it, right? Now towards the right you can see all the labeling in detail: stroma, the chloroplast DNA, the granum, external membrane, internal membrane space, or intermembrane space, internal membrane, plastoglobule, which we can see here, yellow color structure, the ribosomes, thylakoid membrane, lumen, that is space inside the thylakoid or the grana part, then the lamellae which connects the grana. Okay. Further, here I have given little bit more of additional information which you read out. So it will help you to understand the different pigments other than the chloroplast, right? So first you can see the left one, plastid. So I put all the different types which are learning in this, so you can understand with the image, because as I always say, biology is a subject with the image, figure, or the diagram, picture. It helps to understand very easily. So you can see the plastid, ethioplast, crystalline prolamellar bodies, then chromoplast, pigment synthesis and storage, chloroplast, photosynthesis lipids, which we already learned, leucoplast, synthesis of fatty acid and amino acid, amyloplast, storage of starch and sugar, elioplast, storage of lipids and oils, and proteinoplast, sites for enzyme activity or protein activity. So these 7 types which are there, you can see the image of them and what is their main function which they carry out, right? Now right side we come to the precise one, chromoplast, chloroplast and leucoplast, which 3 we are learning in particular. Chloroplast contain chlorophyll, green pigment that absorbs sunlight for photosynthesis, which we are already learned, produce and store glucose. Chromoplast contain carotenoids, that is red and orange color pigment, red, orange and yellow, all 3 pigments which gives coloration to the fruits and flowers, found in flowers and fruits. Leucoplast contain no pigment, so it is used to store starch, as just now we read an example, potato, right? Now coming to the next one, that is ribosomes. Now first you can see the right side, George E. Palade. Okay, now Palade, the scientist who first observed the ribosomes, right? So I put the image so as to see who is the scientist. As you can see in most of the chapters, wherever the scientist name has occurred, I put their images also, so as to know who is the scientist, right? Know the scientist. And also, as I mentioned, the scientists, their names, they always asked in MCQ and NEET questions. At least 3 to 4 scientists name, discovery, what they have done, their Nobel Prize, all those things. So these things which will help you out. Okay. Ribosomes. Ribosomes are protein factories of the cell. They use the genetic information to synthesize proteins. So as we know, main function is synthesis of proteins. Ribosomes were first observed as dense particles in electron micrograph of a cell by scientist Plastids in 1953. Ribosomes are made up of ribosomal RNA and proteins. They do not have any membranous covering around them. So there is no covering as such as you saw in case of mitochondria, chloroplast, etc. In eukaryotic cell, ribosomes are present in mitochondria, plastids and cytosol. Ribosomes in cytosol or cytoplasm are either found attached to the outer surface of rough endoplasmic reticulum and nuclear membrane or freely suspended in cytoplasm. That's why because of the ribosome attached to the endoplasmic reticulum, we use the term rough endoplasmic reticulum. So it is attached to ER or to the nuclear membrane, or it is freely suspended in the cytoplasm. Both are similar in structure and are AT-S type. Each ribosome is made up of two subunits, large and the smaller subunit. Bound ribosomes generally produce proteins that are transported outside the cell after processing in ER and Golgi body. Example, bound ribosome or echinus cells of pancreas produce pancreatic digestive enzymes. So you can see here it also helps in the production of pancreatic digestive enzymes in pancreas, that is echinus cells, which are called bound ribosomes. Free ribosomes come together as and form chains called polyribosomes for protein synthesis. Even this we can see in the further slide in the animation images which I put in this video. Free ribosomes generally produce enzymatic proteins that are used in cytoplasm like enzymes required for breakdown of sugar. Both type of ribosomes can interchange position and even the function. Number of ribosomes is high in cells actively engaged in protein synthesis. As you know, for protein synthesis, ribosomes is necessary. That is why you can easily understand other way around. Ribosomes are high in cells where protein synthesis are taking place actively. Now here you can see the animation image. Two images I put. One you can see the polyribosome, the chain-like structure very clearly, right? Then right side you can see the ribosomes, or you can see the amino acids very clearly. The tmRNA, where you can see the AUG, the codon which attaches to the complementary one UAC, similarly CUA to GAU, where GGC to CCG, and so on it is continuing. So here I just put this animation to understand how the coding takes place or pairing up takes place between the amino acids. Now, always remember, little bit more additional information. As I just mentioned, I put the scientist images, their discovery, invention, what they have done, etc. As I told, this will be always helpful to know the scientist, also their year, work done, what they have done, and any other achievements. So, which always is asked in the NEET examination. Okay, so always remember particle size of ribosomes is measured in terms of Speedberg constant. Yes, as we learned the centrifugation, it is a measurement of sedimentation rate of a particle in ultracentrifuge. It is thus a measure of density and size of particles. So we can learn as 1s as 10 raised to 13 second. So we can see here Speedberg, or we can see Theodor Speedberg, the image which is given by S. The large and small subunit of ribosomes are named according to the velocity of sedimentation when subject to centrifugal force. The unit to measure sedimentation velocity is Speedberg, that is S. The large value faster than sedimentation velocity S, larger the molecule. Named after the inventor of ultracentrifuge, that is Theodor Speedberg. The sedimentation velocity of a function of a particle's weighing or volume or size and shape. Further, know the scientist Venkatraman Ramakrishnan won Nobel Prize in Chemistry in the year 2009, almost around 12 years back, for explaining the structure and working of ribosomes. He shared the prize with Jonath that is who is a scientist from Israel, and Thomas States of USA. Again, I have put the image. Venkatraman Ramakrishnan, United Kingdom, who was born and brought up over there. Thomas A. States, USA, Ada E. Yonath, Israel. So all the three who got the Nobel Prize in Chemistry. Then nucleus, the next organelle. Structure of nucleus of eukaryotic cell becomes distinct in non-dividing cell or during interphase, becomes very clear. Such interphase nucleus is made up of nuclear envelope, nucleoplasm, nucleus and chromatin reticulum. We can say four parts of nucleus. First one, the nuclear envelope, which I highlighted, is a double wall delimiting membrane of nucleus. Two membranes are separated from each other by perinuclear space 10 to 50 nanometer. Outer membrane is connected with endoplasmic reticulum at places. It also harbors ribosomes on it. The inner membrane is lined by nuclear lamina, a network of protein fibers that helps in maintaining shape of the nucleus. Two membranes along with perinuclear space help in separating nucleoplasm from cytoplasm. So as nucleus has its own matrix, nucleoplasm, and the cytoplasm of the cell, they are separating from each other, but the materials may be exchanged between them through the nuclear pore. However, nuclear membrane is not continuous. At places there are small openings called nuclear pores. The nuclear pores are guarded by pore complex which regulate flow of substances from nucleus to cytoplasm and in reverse direction. That is in two or four, whichever components exchange of materials takes place. Nucleoplasm of karyolymph contains various substances like nucleic acids, protein molecules, minerals and salts. It contains chromatin network and nucleolus. Nucleolus is another component which is not bound by cell membrane. Nucleolus is made up of tRNA and the ribosomal proteins, and it is best known as the site of ribosome biogenesis. What is biogenesis? As you know, bio, life, genesis, formation. So we can say formation of ribosomes or the ribosome proteins. Depending on synthesis activity of a cell, there are one or more nucleoli present in the nucleoplasm. For example, cells of oocyte contain large nucleolus, whereas sperm cell contain small inconspicuous one. They appear as dense spherical bodies present near chromatin network. They produce rDNA, that is ribosomal DNA, and ribosomal proteins, which are then transported into cytoplasm and are assembled together to form ribosome. Just as I mentioned, the material exchange may take place between the nucleoplasm and the cytoplasm, by which the ribosomes are formed later on. Then again I put images to understand the nucleus structure. So on to the right, the picture of nucleus, where nuclear pore, nucleolus, nucleoplasm and nuclear envelope, the four parts, right? And then towards the left you can see the animation, slow animation we can say, where the nucleus, the inner nuclear nucleolus, which you can see, the nucleoplasm you can see, where it is slowly moving in the inner component, and onto the sides the mitochondria also you can see, the endoplasmic reticulum and few lysosomes, etc. But main what you have to observe is the nucleus with the nucleolus, nucleoplasm, nuclear envelope, the outer covering. Nuclear pore is not visible as such clearly. Okay. Now, chromatin reticulum, which is present in the nucleus. Nucleus contain genetic information in the form of chromosomes, which are nothing but DNA molecule associated with proteins. In a non-dividing cell, the chromosome appear as thread-like network and cannot be identified individually. This network is called as chromatin material, which you can see towards the right side image, the thread-like structure. The chromatin material contains DNA, histone and non-histone proteins and RNA. In some regions of chromatin, DNA is more and is genetically active, called euchromatin, or we can say U, the term is given to two chromatin material. Some regions that contain more of protein and less of DNA are genetically inert and called heterochromatin. So here when DNA is more genetically active, euchromatin, where DNA is less, protein is more, it is less, it is genetically inert, heterochromatin. So two types, euchromatin and heterochromatin. When the cell prepares to divide, the chromosomes coil and get condensed. At metaphase stage, they become distinct and can be clearly identified. That's why out of the four phases, prophase, metaphase, anaphase, telophase, the clear phase is the metaphase, more in comparison to the other three phases. Or in all the four phases, metaphase is a very clear and distinct phase. Every species of living organism has specific number of chromosomes, like normal human cell has 46. The nucleus contains entire genetic information, hence play important role in heredity and variation. In this, it is a site for synthesis of DNA, RNA and ribosomes. So here what is the synthesis taking place for DNA, RNA and ribosome, all the three. It plays an important role in protein synthesis also. Chromosomes number being constant for a species, it is important phylogenetic studies. Now what does this mean? Now for example, human being, for us what is the number of chromosome present? It is 23 pairs. So in each of these species it is constant. And what is our main terminology here? Phylogenetic study. So it can help in having the phylogenetic relationship from our earlier genes, earlier DNA which was present, or the evolutionary studies can be done. Nucleus thus is the master cell of organelle. That is, we can say it is a main cell, important cell, or we can say if in the house, the main power is when you enter the house, leave the house, the electricity, which is the important component, right? So here in case of cell, the nucleus being the master cell organelle. Okay, then next, cytoskeleton. With advancement in light and electron microscopy, scientists revealed presence of network of fibrils through the cytoplasm. It is called as cytoskeleton. Cytoskeleton consists of microtubule, microfilaments and intermediate filaments. So three parts are there. Now repeat the terms: microtubules, microfilament and intermediate filament. Now take one by one. Microtubules, they are made up of protein, tubulin. Microfilaments are made up of actin and intermediate filaments are composed of fibrous proteins. Cytoskeleton helps in maintenance of shape of cell, contraction of cell, mobility of cell and cell organelle shape, changes in shape of the cell and cell division. So you can see what is the importance of cytoskeleton: to maintain the shape, to contraction of the cell, mobility of the cell and cell organelles, to change of the shape of the cells and also in cell division. Here also I given a few images along with the cytoskeleton, that is direction of motion, that is for flagella, direction of motion which is carried out by the flagella, and in the second one the cilia. First one, the example is euglena, which has got a single flagella, and the lower one paramecium, which all of you know, which has got cilia, which is slipper like body, cilia present throughout the body, right? And the right side you can see the cytoskeleton with the various parts: plasma membrane, central microtubules, radial spokes, inner sheath, next fin, B tubule, A tubule is doublet, microtubule, dynenums, etc. Then cilia and flagella. Again, what we saw the image of euglena and paramecium. Here I had a very good animation image. So on the right side you can see the animation of cilia, how it is moving, which helps in the movement of paramecium, and lower part towards the right, flagella, which helps in the movement of euglena, right? So coming to the points, the fine hair-like membrane-bound protoplasm outgrowth that occurs on the free surface of the cell. They generate a current in the fluid medium for passage of material and locomotion, which you can see in the image. Cilia are small in size and many in number. Cilia act as a or causing movement of cell, which you can see in paramecium. They are numerous, small, help in the movement. Whereas flagella, they are usually long and few in number. They may be 1, 2 or 3 in number, but they are long in comparison to cilia. Flagella present in prokaryotic bacteria are structurally different from that of eukaryotic flagella. So that both are different in prokaryotic and eukaryotic. Cilium or flagellum consists of basal body, basal plate and shaft. Three parts are present in both. Basal body is placed in the outer part of cytoplasm. It is derived from the centriole. It has got 9 peripheral triplets of fibrils. Shaft is exposed part of cilia or flagella. It consists of two parts, sheath and axonema. Sheath is covered membrane of cilium or flagellum. Sheath is covering membrane of cilium or flagellum. Core called as axonema possesses 11 fibrils running parallel to the long axis. It shows 9 peripheral, means towards the outer lining, and 2 central or central fibers. So the arrangement is 9 plus 2. Now this particular image you can see in the next slide. The central tubules are enclosed by central sheath. This sheath is connected to one of the tubules of peripheral doublets by a radial spoke. Central tubules are connected to each other by bridges. The peripheral doublets are connected to each other through linkers of interdoublet bridge. Now you can see the image. We'll go to the points first. Centrosome is usually found near the nucleus of an animal cell. It contains a pair of cylindrical structure called centrioles. The cylinders are perpendicular to each other and are surrounded by amorphous substances called pericentriolar material. Each cylinder of centriole is made up of 9 set of triplet microtubule made up of tubulin, which you can very clearly see in the image. Evenly spaced triplets are connected to each other by means of non-tubulin proteins. At the proximal end of the centriole, there is a set of tubules called hub. The peripheral triplets are connected to the hub by means of radial spokes. Due to this, proximal end of centriole looks like a cartwheel, which you can see in the image. The centrosome helps in assembly of spindle apparatus during cell division. It forms basal body of cilia and flagella. So you can see the image. The first part, you can see microtubular triplet connecting fibers. In the second, which appears like a spoke or the wheel-like appearance, a cylinder, foot, radial fiber, microtubule, which is triplet. We can see in threes. In both the image, we can see the structure both whether it is horizontal or vertical image. Okay, so that ends with the chapter and today's third part, and also the questions and assignment based on this third part. Okay, thank you all of you.